After our discussions on the amino acids, their properties and the formation of the peptide bond, we now go on to see what this polypeptide chain entails in terms of its structure. So, our first description here is going to be on the primary structure of the proteins in our second module, that is going to be concerned with the protein architecture. When we consider the primary structure, before we go into an understanding of what the primary structure is or what the amino acid sequence is, we have to understand a bit about the geometry of the backbone, as it is called. We understood that when we have the peptides linked together, we have the peptide bond itself that has a partial double bond character with restricted rotation. But in addition to the single bonds, the other single bonds that are present, there is a possibility of rotation, and because of this rotation there are various factors that play an important role in protein architecture. When we look at the three-dimensional conformation of proteins, the amino acid sequence, the N-terminal, the torsional angles, and also the mass to charge ratio in terms of the total protein and how we can go for specific sequencing methods. And we will understand the necessity of doing this or understanding this because we want to know more about the proteins, the protein chemistry, the protein function, and so on. So, the first thing that we can think of what is proteins, what are proteins? Proteins can be of varying types. Okay, they can be enzymes, they can be hormones, they have structural proteins or even antibodies. So, if we look at the overall structure of the proteins, they are distinct in nature. They are composed of the common amino acids, the ones that we studied in the previous module, but they are linked together in varying fashions, they fold in varying fashions, and because of that they result in different types of three-dimensional structures that lead to different types of functions, and this is what we will gradually understand as we go through the course. So, if we look at proteins such as trypsin, that is shown here, insulin and immunoglobulin, which is an antibody. Now, trying to understand this, we will try and realize what these particular representations mean. And why do we have these representations? Why is this folded structure like this in terms of its interactions? An extremely important factor in trying to understand what we mean by protein folding. Now, when we consider the protein structure, there are different levels. The primary structure is one such level. What do we have for these proteins? As we saw in the class for the peptide bond, we had the formation of a polypeptide chain. Now, what this polypeptide chain does is it has a combination of multiple amino acids that form a sequence, and the interaction with each other follows or finally forms a three-dimensional structure. For now, we know that we have a polypeptide chain linked by peptide bonds. That is what forms the primary structure. After this, we have the secondary structure, the tertiary structure, and the quaternary structure that is formed by some proteins, which we will see as we go along in the lectures. When we look at the primary structure of proteins, as we consider, they consist of the sequence of amino acids, as we found out, that were linked by the peptide bonds. So, this is our sequence of amino acids. What we know is on the left hand side we have the N terminus, on the right hand side we have the carboxyl terminus, because we know that this peptide bond is formed from a combination of the CO and the NH. Now, if we look at the way this peptide bond has been drawn, the N terminus is this one. The C terminus is this one. So, this particular R group is amino acid number 1. This is amino acid number 2. This should not lead to any confusion whatsoever. The amino acid that begins the protein is the N terminus. We know that how is the peptide bond formed? It is formed from the COOH of amino acid number 1, say, that is going to condense with amino acid number 2, say. And what do we have? We have the loss of this water, as we saw in the lecture on the peptide bond, and we have the C double bond O NH form. The C double bond O and the NH that is marked here, the peptide bond. But we realize that this C double bond O has to be from the previous amino acid. So, this is the direction of the N terminus and this is the direction of the C terminus. And you have to know how to identify the directionality of the polypeptide chain, because it has a definite direction to it. So, the primary structure consists of the sequence of peptides, sequence of amino acids linked by peptide bonds. The other covalent links that are present in the polypeptide chain are the disulfide bonds. We saw this when we studied the cysteine, where we had the S bond formation from 2 cysteine amino acids. When 2 cysteine amino acids link together to form the disulfide linkage in proteins, it is called a cysteine, and this is helpful in bringing a folded structure in the terms of having different parts of the polypeptide chain link together. This may be cysteine number, say, 58, cysteine number 58. This may be cysteine number 102 in a large protein. So, what happens is when they come together, they bring different parts of the protein close to one another in 3-dimensional space, which we will see later on. So, the covalent bonds, apart from post-translational modifications, which I had mentioned earlier, these are the other covalent linkages that are present in proteins, the disulfide bonds, in addition to the peptide linkages that we studied. So, here is the cysteine that I was talking of, the cysteine, and we look at. So, this is where we have the formation of the disulfide, 2 cysteines coming together forming a disulfide linkage, a covalent linkage. Now, when we look at the amino acids themselves, if we just go back a slide and look at the amino acids themselves, there are several single bonds. If we have a longer polypeptide chain, we have several single bonds about which we can have rotations. Now, apart from this, the amino acids themselves, the ones that have longer chains, what do we mean by that? Suppose we looked at the nomenclatures, we have a C alpha followed by a C beta, followed by a C gamma, even a C delta, and so on and so on. Now, all of these are linked by single bonds. So, there is a possibility of rotations about these bonds. This gives what are called rotamers, giving us chi angles. So, the rotational isomers, we saw the different kinds of structural isomers that we can have in terms of a cis trans for the peptide. But when we look at the geometry, it is important to understand that the amino acids themselves, because of the presence of the single bonds, can have specific side chains that can rotate, because we have single bonds that link the carbons together. So, if we look at the torsional angles that describe the dihedral rotation, there are specific dihedral rotations which we will study in a minute, very important for the protein backbone geometry, and these are the side chain rotamers that give a library of possible structures because of the rotation along the chi1, chi2, chi3. What do we mean by this? This is our amino acid where we have a peptide bond. So, this is our peptide bond, C double bond NH. So, this is our peptide bond. Now, we are looking at a definition for a dihedral angle. Now, a dihedral angle is an angle present between two planes. So, for this we have to define, say, 4 atoms. In a sense, if we have 4 atoms linked together, A, B, C, D, we know that we can define a plane by using just 3 points or 3 atoms. So, if I have these 3 atoms, I can define a plane using ABC. I can define another plane using BCD, and the angle between these 2 planes is what we call the dihedral angle. So, if we want to look at it differently, say we have A positioned here, B, C and D. So, if I look at a plane that comprises A, B, C, and I look at another plane that comprises B, C, D. So, this is the other plane that we see that has B, C, D. So, if we look at the angle, this particular angle, that this is our dihedral angle. So, depending on the definition, it will be phi, psi, omega angles that we will see as we go along. So, the dihedral rotation is in terms of the backbone angles, phi, psi, omega. But when we look at the side chain, as we looked at the different single bonds, so this is the arginine amino acid residue, and we see there are several single bonds. Now, there is rotation possible about these single bonds, which is what is termed as the chi1, chi2, chi3, giving us the rotamers that usually represent a local energy minimum in terms of the torsional angles, giving us the best possible structure or the best possible orientation that is going to give us the minimum energy. The important method to construct a rotamer library is what is called a cluster approach. Given that we have the C single bond linkages, the carbon atoms are sp3 hybridized, which means that if we have a connectivity between the C alpha and the C beta followed by the C gamma, we can have rotations about these angles as we saw in the previous slide. Now, these rotations give us angles of chi1, that is a rotation about the C alpha, C beta, a chi2 that is around the C beta, C gamma, and a C gamma, C delta is the chi3. Now, because of the others, so this is our C alpha and then we have the C beta, C gamma. This is where we have the nitrogen that is part of the backbone of the amino acid residue. Now, when we look at these specific rotations, we see the C beta here that is in turn connected to the C alpha. So, depending upon the geometry, there can be specific orientations possible so that there are no steric clashes of the atoms coming together or the atoms bumping into each other. So this is where we have specific geometrical constraints depending upon the structural characteristics of the amino acid side chains, the chi1, chi2, chi3 angles. But if we go a step further in understanding the types of interactions that are used to find the conformations of protein side chains with the repulsive steric interactions that are possible, this is what is important. So we have some that are backbone conformation dependent. We have, these are the chi1, chi2, where we have these linkages that we were looking at. Then we have the backbone, these are independent of the backbone conformation, these are dependent on the backbone conformation, because this will comprise the phi, psi and the chi1, where Ci minus 1 is the carbonyl carbon of the previous amino acid. We will go into the geometry a bit more because we are looking at protein architecture. So we have a specific model that defines this. But now when we look at the phi psi angles, there each of these amino acids has two main chain backbones defining dihedral angles phi and psi, and we know already that the peptide bonds have restricted rotation because of the partial double bond character that is represented by this dotted line that you see here, where we have the CO and the NH. Now when we connect the others, so this is our C alpha, so this is the C alpha and we can see this as the side chain. Now what happens is rotation about this angle, rotation about this bond rather, and rotation about this bond is also possible, giving us the dihedral angles that are extremely important in the protein geometry. What we have here is the C alpha carbon, we have the C, this is the C double bond O here, this is the nitrogen and this is the oxygen. So this is what is important in understanding the protein geometry. So when we look at the phi angle, we will see that this is what the dihedral looks like. So now when we rotate this, we can see that we can have a 90-degree phi angle. So the first thing that we saw that there were 4 atoms, 1, 2, 3, 4, and we are rotating the, so we are going 1, 2, 3, 4. When 1 and 4 clash, then we have, so this is what we are looking at. We are looking along 2 and 3 and we are looking at a rotation along 2 and 3, and we have a clockwise rotation that gives us 0 to 180. We can also have a anti-clockwise rotation that will give us a minus 180. So this gives us what is the phi angle, that is the angle between the 4 atoms that have to be defined are the C alpha, the C, the N and the O. So that is what is going to define our geometry for the phi angle. We similarly see a geometry for the psi angle that is also going to give us the factor from the specific portions that we looked at in defining the backbone atoms. So the function of a protein is going to depend upon its amino acid sequence, and the most important part here is the primary structure, which is the link of the different amino acids one after the other in forming the polypeptide chain. Each protein has a distinctive number and sequence of amino acid residues, and given that there is no limit to the number of amino acid residues that you can use in forming this polypeptide chain, you realize that there can be very many different proteins that can be formed. So the primary structure of a protein depends on how it actually forms its unique 3-dimensional structure that ultimately determines its function. We looked at the proteins with the different functions always have different amino acid sequences, but the overall structure sometimes is pretty much the same. We looked at how the variation in a single change of amino acid from glutamic acid to valine could result from a normal red blood cell to a sickle cell, depending on the change or the mutation in the type of amino acid that we are talking about, because we are changing the characteristics. Similarly, when we have muscular dystrophy, there is a deletion of a large portion of a specific polypeptide chain leading to impaired function. So, we realize the importance of the primary structure, the importance of the sequence of amino acid residues in the specific way they form, because that is going to be dependent on how they fold in three-dimensional space. Now, if we want to look at the amino acid sequence, there are methodologies that help us in determining the sequence of the proteins. Now, if we look at insulin, this is the amino acid sequence of bovine insulin, and we find some specific features here. There are two chains in insulin, the A chain and the B chain. Let us try and look at the structure and see what we can understand from this. We have a disulfide linkage in this case in the A chain, that is intra chain, meaning it is in chain A itself. We have in addition two disulfide linkages that link chain A and chain B. Now, if we want to understand or we want to know what the sequence of chain A is, what the sequence of chain B is, then there are specific methodologies that have to be followed in order for us to identify the specific amino acids that are linked in the specific order that forms what the A chain is or what the B chain is, or for that matter whatever protein it is. So, an understanding of that sequence is extremely important, because that is what defines its function. So, just for a preliminary idea to understand Sanger's method for peptide end group analysis, it is just a labeling. So, if we have a methodology by which we can label the N terminus of the polypeptide chain and we can cleave it, means we can break it off from the rest of the polypeptide chain and have a methodology to analyze which amino acid has been broken off, I can find out what the N terminus is. So, this is the methodology that is commonly used, and we will see how we can actually use these for understanding the sequence. There are very interesting methods, more like puzzles and riddles, that actually tell us or give us an interesting way into finding out what this sequence is all about. What do we want to know? We want to know what is R1, what is R2 and what is R3. So, the method in this case is using a reagent, dinitrofluorobenzene, and there is a specific reaction that occurs. After this reaction occurs, we have the formation of this adduct. Now, this means that there is the R1, R2, R3 in the polypeptide chain are there, and we now have this product formed from the reaction with DNFB. Now, when we have hydrolysis, what happens is the hydrolysis results in the breakage of this bond. Like we looked at last time, when we have condensation, we have the formation of the peptide bond. When we have hydrolysis, we can have the disruption of the peptide bond. Now, when we have the disruption of the peptide bond, what we are going to see is we are going to see a series of amino acids. But the information that we get is we have to look for which amino acid is tagged along with the DNFB, and this would be amino acid number 1. So, this is one method in which we can determine this. Another method is known as Edman degradation, which forms what is called the phenyl thiohydantoin derivative. So, here we have the peptide, here we have a specific reagent that comes into the picture where we have a reaction. This reaction occurs at a specific pH, giving us again a derivative that is known as the phenyl thiohydrantoin derivative, and this reaction then gives us the peptide along with the first amino acid of the sequence, and this is important in an understanding of what the identifier is for the first amino acid, because this is the rest of the peptide. Now, the interesting part here is that we can now subject the rest of the peptide to the same reaction, and because now we have a new N terminus. So, we can find out what that N terminus is based on the specific sequence of the reaction. So, in this degradation, we can find out the N terminus. We want to know the N terminus, meaning we want to know what amino acid is there, what R group is present there. So, this is how the specific reactions occur in the Sanger's method and the Edmond degradation. We will look at specific methods to find out the sequence of polypeptides in a further lecture later on. Another way in which we can find out this is using mass spectrometry, where we can provide the sequences of multiple short polypeptide segments in a protein sample quite rapidly, where we have actually a fragmentation. What happens in this case is, in mass spectrometry, we have different methods. We have MALDI-MS, that's matrix assisted laser desorption. We have ESI, electrospray, and we have also MSMS. Now, these are different mass spectrometry methods that tell us or give us an idea as to how we can determine the sequence. So, in this case, the analytes are ionized in vacuum. So, we have different fragments that are formed, and these fragments are now charged. Now, because of the newly charged molecules, they are put into an electric field and they migrate along this electric and magnetic field because of their mass to charge ratio. So, this is what we see in an electrospray ionization MS data, a fragmentation of the polypeptide chain. Similarly, if we look at a tandem mass spectroscopy, we have enzyme and chemical fragmentation, which means that if we have a sequence of amino acids, what we do is we have a sequence, we can break it off enzymatically, and then we analyze the different fragments that we have, and this analysis can be done through tandem mass data or tandem mass spectrometry, where we can look at a specific sequence of the polypeptide chain, and that is extremely important in an understanding of the primary structure of the protein. So, we looked at what the primary structure of the protein is, the sequence of the amino acids, the dihedral angles and the rotamers, dihedral angles, the backbone angles of the amino acids, which we will revisit when we look at the secondary structure, the rotamers for the amino acids themselves, and specific amino acid sequencing methods, giving us an idea of what the primary structure of the protein is. Thank you.